Welcome to Practical Biochemistry. Today we are going to perform test for identification of proteins in a given sample. First of all, let's talk about proteins. Proteins are large biomolecules or macromolecules that are comprised of one or more long chains of amino acids. Amino acids are organic compounds that contain amino and carboxyl functional groups, along with a side chain specific to each amino acid. The key elements of an amino acid are carbon, hydrogen, oxygen, and nitrogen, although other elements are found in the side chains of certain amino acids. Proteins differ from one another primarily in their sequence of amino acids, which is dictated by the nucleotide sequence of their genes, and which usually results in protein folding into a specific 3D structure that determines its activity. The individual amino acid residues are bonded together by peptide bonds and adjacent amino acid residues. The sequence of amino acid residues in a protein is defined by the sequence of a gene, which is encoded in the genetic code. Some proteins have non-peptide groups attached, which can be called prosthetic groups or cofactors. Like other biological macromolecules such as polysaccharides and nucleic acids, proteins are essential parts of organisms and participate in virtually every process within cells. Many proteins are enzymes that catalyze biochemical reactions and are vital to metabolism. Proteins also have structural or mechanical functions, such as actin and myosin in muscle and the proteins in the cytoskeleton, which form a system of scaffolding that maintains cell shape. Other proteins are important in cell signaling, immune responses, cell adhesion, and the cell cycle. In animals, proteins are needed in the diet to provide the essential amino acids that cannot be synthesized. In this practical, we are going to test the presence of amino acid by performing nitric acid test, biuret test, and xanthoprotease test. The materials used in the following test for proteins are concentrated nitric acid, sodium hydroxide solution, copper sulfate solution, test tube and test tube holder. Nitric acid test for amino acids. In nitric acid test, we use concentrated nitric acid for testing the presence of protein in the given sample. Procedure: take 3 ml of concentrated nitric acid in a test tube and then add the given solution to the test tube in inclined position, allowing the solution to run down the side of the tube. A yellow-colored ring is observed at the zone of contact of protein solution and nitric acid. This thus proves the presence of amino acids. Xanthoprotease test. Xanthoprotease test is used to differentiate aromatic amino acids which give positive result from other amino acids. Xanthoprotease test is used to detect amino acids containing an aromatic nucleus in a protein solution which gives yellow color nitro derivatives on heating with concentrated HNO3. The aromatic amino acids are tyrosine, tryptophan, and phenylalanine. The aromatic benzene ring undergoes nitration to give yellow-colored product. Phenylalanine gives negative or weakly positive reaction, though this amino acid contains aromatic nucleus because it is difficult to nitrate under normal condition. Procedure: take 2 ml of amino acid sample in a test tube. Then add 0.5 ml of concentrated nitric acid to the test tube and then heat solution. The solution then appears to be yellow-colored. The above solution was then cooled under tap water and 2 ml of sodium hydroxide solution was added to make it distinctly alkaline. Here we can observe that the yellow color of mixture was changed to orange. This thus proves that the given sample solution contains aromatic amino acids. In presence of concentrated nitric acid, the aromatic phenyl ring is nitrated to give yellow-colored nitro derivatives, and at alkaline pH, color changes to orange due to ionization of phenolic ring. Let's perform biuret test. Biuret test is a general test for compounds having a peptide bond. This test is given by compounds containing 2 or more peptide bonds. Since all proteins and peptides possessing at least 2 peptide linkage, that is tripeptide, gives positive biuret test. The principle of biuret test is conveniently used to detect the presence of proteins in biological fluids. Alkaline CuSO4 reacts with compounds containing 2 or more peptide bonds to give a violet-colored product, which is due to formation of coordination complex of cupric ions with unshared electron pairs of peptide nitrogen and oxygen of water. Procedure of biuret test: take 3 ml of diluted sample solution in a test tube, then add equal volume of 10% sodium hydroxide solution. Yellow color was observed which was then mixed thoroughly and 0.5% copper sulfate solution was added drop by mixing between drops. The appearance of blue color proves negative biuret test and the appearance of purple color Proves presence of peptide bond, that is presence of amino acids. Thanks for watching. Please like and subscribe for more similar contents.